Okay, so now we know how enzymes work. Let's look at how environmental conditions are going to impact our enzymes and how well they work. So remember that your body is always trying to maintain homeostasis. And what this is basically maintaining constant internal conditions. And the reason why this is important is because all of these things can influence how well enzymes work. And if they change too much, if the conditions within your body change too much, your enzymes won't work anymore and you won't be able to carry out all the metabolic reactions that basically keep you alive. Which is why getting a really high fever is a really, really bad thing because it's going to change temperature too much. Okay, so the first thing we're going to look at is temperature. Okay, so let's look at temperature first. So here is a graph showing enzyme activity, and you can record this in various ways. It might be how much substrate is converted to product, for example. And down here is the temperature, and this is a degrees Celsius scale. So this is going to be boiling, and this is freezing. So if we plot how well enzymes work under different temperatures, we're going to see a graph like this. Okay, so when it's very, very cold, enzymes won't really work. And as the temperature increases, we are going to see an increase in the rate of reaction. And the reason why is because the hotter molecules are, the higher the temperature, there's more kinetic energy. And the higher the kinetic energy, the more substrates are going to collide with the active site. Because the way enzymes work is the substrates aren't moving into the active site deliberately. What's happening is you've got enzymes mixed with substrates, and all of these molecules are moving around. And as they move around, they collide with one another, and the substrate enters the active site. So the faster these molecules are moving, the more substrates are going to enter the active site and the more reactions will take place. So when we get to this point here, this is the optimum temperature for our enzymes to work. They're going to work really, really well. Then what we see happening is we see a really strong decline in enzyme activity to nothing. And the reason why, what's happening here, is that the amino acids within our enzyme, they've got kinetic energy as well. And so what's happening is that they are moving within the enzyme. Okay, the monomers of the polymer are moving around. And what's going to happen is that if they move around too much, is that the bonds are going to get broken. And that is going to change the shape. If we change the 3D shape, the substrate cannot bind. And then we say that the enzyme has been denatured. So to denature an enzyme means to change the three-dimensional shape. If we change the three-dimensional shape, the substrate will no longer fit. So our substrate cannot fit within this active site here, and the reaction can't occur. So that is how temperature is going to influence our rate of enzyme activity. Now let's look at pH. Now the way pH is going to work is, here's our enzyme here. The way that pH is going to work is it's going to influence the bonds between our amino acids. So if you remember, our enzyme is a protein, which is a polypeptide chain, folded up, and there are bonds holding the folded up shape together. Okay, so what happens is that if we change the pH, these bonds here are going to get broken. and we are going to denature our enzyme. So the three-dimensional shape is going to change and it cannot bind with the substrate. Okay, so that is how pH is going to influence our rate of reactions. Okay, now something really interesting is that if we look at the rate of a reaction, enzyme activity, and we look at pH here, where we've got high pH, which will be 14 on our scale, and here we've got low pH, which will be 1. So acidic down here and alkali up here. We're going to see different pHs being optimal for different enzymes. And the reason why is because, well, you've got lots of different conditions within your body. So here, this might be in your stomach. So some of the enzymes like pepsin, for example, that breaks down proteins in your stomach, the conditions in your stomach are very acidic, and so your enzyme has to work in those conditions, otherwise you wouldn't be able to break down proteins in your stomach as you digest your food. Here might be in the mouth, where conditions are more alkaline. Okay, so this might be, for example, salivary amylase, which breaks down starch. Okay, so this enzyme here is not going to work inside the stomach. Now, as far as being denatured, when we're seeing this thing happening here, some enzymes are permanently denatured by pH, some are just temporarily denatured by pH. So what can actually happen is that if you then return the pH back to optimal conditions again, your enzymes will resume working, these bonds will reform, and the enzyme will work again. All right, now let's look at these last two things. So we're going to look at substrate concentration, and we're going to look at enzyme concentration. Okay, so remember that enzymes are not used up in our reactions. So let's draw a bunch of enzymes. Okay, so here are our enzymes, and then let's draw some substrate. Now, right now we have 1, 2, 3, 4, 5, 6 enzyme molecules. Okay, so all of these substrates combined within the active site of these enzymes, and these reactions can occur. However, if we really increase the substrate concentration now, are we going to get a faster rate of reaction? The answer is going to be no, because these enzymes can only work with one substrate at a time. So in this situation, if we've got a limited number of enzymes, we are going to have a limited rate of reaction. Okay, so the enzyme concentration here is limiting how fast the reaction occurs as a whole. So increasing the substrate concentration here isn't really going to do anything. Okay. However, let's say we increase the enzyme concentration. Okay. And I'm just going to get rid of some substrate. So now we have got more enzyme than substrate. So now if we increased the substrate concentration, it would actually make a difference to our rate of reaction, okay, because we've got extra enzymes available to be catalyzing our reactions. Okay, so substrate concentration and enzyme concentration kind of work together to influence the rate of reaction. And you're going to get to a point where, well, if you've got not enough enzymes for the substrate, it's not going to make any difference. Now, something you're going to encounter in the lab is surface area. So I'm going to want you to think, let's look at this area over here. You've got something that has got enzyme in. For example, when you do your lab, you're going to look at potato catalase, and how much potato you've got is going to influence how much catalase is there, but also the surface area. So I want you to be thinking, what has a greater surface area? Let's say you've got a piece of potato that's 1 gram. What's going to expose more enzymes to the substrate? This shape here, or for example, if you got 1 gram of potato in lots of strips like this, or for example, diced up your potato into lots of little squares or mashed it up, what's going to expose more enzymes to more substrate? So that's some of the things I want you to be thinking about.